Hello everyone. My name is Harshpatki from SYBSc Microbiology. Today I'm going to present a presentation on investigation of biomolecular structures, its function and analytical techniques. So let us understand what actually these biomolecules are. So, what is biomolecules? Biomolecules are organic molecules which are important and essential part of life. Without biomolecules, life wouldn't exist. So, these are organic molecules, and there are different types of biomolecules, for example, carbohydrate, lipids, nucleic acids, proteins, which are biomolecules. And they are essential and they are performing structural, functional, and regulatory roles in the cells. So, whatever reactions are taking place inside the cells are because of these biomolecules, and hence the cell is able to perform different kind of activities throughout the day. What activities we are performing on cellular level are because of the interactions and the reactions which are taking place between these biomolecules. So, the types of biomolecules which we are going to study are carbohydrates, then we have protein, we have nucleic acids, and we have lipids. So, there are different types of biomolecules which you can see, that one is the carbohydrate, one is the nucleic acid, which you can see. There are lipids, there are proteins. These all are different types of biomolecules which are existing. So today my project is on nucleic acid. I'm going to explain you about nucleic acid, a basic info about nucleic acid. So nucleic acid are complex biomolecules made of nucleotides. So there are different types of nucleotides which are capable of storing the genetic information. They're going to store the genetic information, they're going to transmit out and express the genetic codes. So, what are the types of nucleic acid? What comes under these nucleic acid? So, there are two types. One is the DNA, one is the RNA. DNA is deoxyribose nucleic acid, and RNA is the ribonucleic acid. So, the structure which you can see here is the nucleic acids and the uh, nucleic acid at the minute level. So, what is the function of these nucleic acid or DNA or RNA, which is a type of nucleic acid? It is helping in protein synthesis, gene regulation, expression of a gene throughout whatever genetic information is there. It is stored inside this nucleic acid. As I said, there are two types. One is the DNA, one is the RNA. So, what is the overview? First of all, the definition of nucleic acids, it is a biomolecule. Again, as I said prior, also it will be storing and transmitting the genetic information. It is a very important type of nucleic acid as it is having the genetic codes for a particular protein which is going to form. I always believe nucleic acids have some relation with protein, as these codes are further going to be taken and it is going to be converted into a protein through amino acids. But what is the structure of nucleic acid? Let's go on to a basic structural level. So, first of all, the nucleic acid is made up of nitrogenous bases. You can see these pink, blue color type of things which you are seeing on the screen is the nitrogenous bases. It is adenine, guanine, cytosine, thymine, and uracil. Here there is U because this is an RNA molecule. But in DNA, this uracil is replaced by thymine. Then we have a pentose sugar, that is a 5 carbon wala sugar. And then we have a phosphate group which is attached at the end and it is connected to the next sugar. So this is the basic structure and it is labeled. So to understand what are the types of uh, nitrogenous bases, as I said, adenine, guanine, cytosine, and thymine, and uracil. And it is mentioned thymine is in DNA and uracil is in the RNA. The adenine and guanine are always termed as purines because they are the double ring structures, while the pyrimidines are the single ring structures, which is nothing but the cytosine, thymine, and uracil. Then we will go on to the pentose sugar, which is there. It is a 5 carbon sugar that is going to form the backbone of the nucleic acid. Every sugar molecule is attached to one phosphate and one nitrogenous basis. So it is very important. It is doing the work of attachment and it is a backbone of the nucleic acid. The sugar which is present inside the DNA will be deoxyribose sugar, right? So these deoxyribose sugar lack an oxygen at the second carbon. At 2 prime carbon, at the second carbon, it is going to lack an oxygen. While in the RNA, where we have ribose sugar, it will have an hydroxyl group, that is OH group at the second carbon. So in DNA, oxygen is lacked at second carbon, and in RNA, OH group is situated at the second carbon of the pentose sugar. And then we have the phosphate group. So you can see this is the nucleotide, then this is the pentose sugar, and this is the phosphate group. So this phosphate group is a very important component of the nucleotide structure. So it is also going to form the backbone. These phosphate group are always attached to the pentose sugar, okay, of the in DNA RNA. So these 3 prime carbon of the deoxyribose sugar in the adjacent nucleotide is going to be present, creating a sugar phosphate backbone. So as we can see that both are attached to each other, there is going to be a sugar phosphate interaction forming a sugar phosphate backbone. Now what is this going to actually do? It is going to give DNA its double helical structure. So 5 prime carbon of deoxyribose sugar will be attached to the phosphate group, which is going to give the double helical structure or the special characteristic structure which we always see that DNA wala structure due to this phosphate group attached to the pentose sugar or deoxyribose sugar. Further, now when I talk about the linkage, so there is always a phosphodiester bond which is formed between the DNA, okay, molecule, and this phosphodiester bond is covalent, right? So there is sharing of electrons which is taking place. So it joins 3 prime, so we have OH, one of the nucleotide sugar, to 5 prime phosphate of the next nucleotide. So at 5 prime we have the phosphate and the sugar. And the yellow color highlighting shows the phosphodiester bond. Again, it is helping in the sugar phosphate backbone of the DNA as well as RNA. Now, these nucleotides bases which are there, it affects the three-dimensional structure of the nucleic acid. As I said, because purines and pyrimidines are weakly basic in nature. Due to the basic nature, they are going to be weak. Purines and pyrimidines have alternating double and single bond. So it is going to show resonance. Okay, that is the partial charges. Then bases can shift form depending on pH. At 7 pH, that is at neutral, certain tautomeric forms dominate. So because of tautomerism, there is an effect in the three-dimensional structure. Then due to the resonance, basis absorbs ultraviolet light So this ultraviolet light which is there at 260 nanometer will be absorbed, right? And then these nucleic acids are detectable by UV spectrometry. So through UV spectrometry we can see, or at neutral pH, these bases which are there are going to be hydrophobic and they will be dissolving in water. Okay, or rule, that is DNA base composition. The base composition of DNA varies from one species to another species. So the RNA which is present is single stranded and it is involved in the protein synthesis. It is a very important structure which is carrying out the genetic information from the DNA, and it is going to help in formation of protein through transcription, translation, and translocation processes through the different types of RNA, that is mRNA, tRNA, rRNA. mRNA is the messenger RNA which will carry out the code for protein synthesis, and rRNA is a component of the ribosome which will move, and tRNA is a transfer RNA which will transfer different amino acids, and then this rRNA will move. The ribosome will move, and all these amino acids will be bonded together by peptide bond, through which the single protein will be formed. Similarly, we have snRNA, that is small nuclear RNA for RNA splicing, miRNA for regulating the gene expression, and siRNA, that is small interfering RNA, which is helping in the RNA interference RNAi technique. Long non-coding RNA for which regulates the gene expression and chromatin structure. That there are different types of genetic information which is being stored in the DNA, which will be passed on from one generation to the another, from parents to offspring. So it is a hereditary information which is going to be transferred from DNA. Then the protein formation which takes place in our body has these codes are present inside the DNA. So by linking different amino acids, a protein will be formed which is necessary for one's life function. There are different proteins for muscles, bones, and for different functions of the life. Now the protein has functional group, sorry, the DNA has functional group, that is amines, ketone, and hydroxyl group, which is present at the nitrogenous bases, phosphate group, and sugar group. So on sugar group we have hydroxyl, we on phosphate we have OH, and nitrogenous basis we have amine and ketone. Then the functions is to store and carry out genetic information during the replication of the cells. This is going to be transferred, and it is a form of inheritance which is going to be transferred. Protein synthesis through components and types of RNA, gene regulation and signaling. And then ways to study are DNA extraction and other methods. So these are the methods through which we can actually perform DNA extraction, and this is one of the photograph through which DNA will be precipitated upwards and then it can be collected, and further it can be taken for polymerase chain reaction PCR technique, which makes multiple copies of a DNA. The steps which are involved are denaturation, that is the DNA strands which are there, they will be separated because we are heating it. Then second will be annealing, that is cooling, where short DNA primers will attach to the target. So target will be attaching the DNA primers, and then we'll be having the extension, that is a warming process, when a special enzyme called as a Taq polymerase will build new DNA strands. So in this way, it is carried out for 35 to 40 times, and a million copies of DNA are produced. So this is the PCR machine through which many DNA copies will be produced. We have agarose gel electrophoresis through which we can actually separate the DNA fragments on the basis of their size. We have to prepare the gel, and we have to pour the sample, we have to load the sample, and then once we are turning on the electricity through which the charge, these DNA will start moving onto the agarose gel. The fragments will migrate towards the positive pole because DNA is itself negative charge. And then we can visualize this DNA under the light, and we can actually uh, observe it by the marked one, and we can actually know the size of the DNA through this method. This is the agarose gel electroporation machine, and this is through which the DNA will run and will be separated on the agarose gel. So thank you for watching, and we can try many more techniques by taking out nucleotide samples. There are many machine like RT-PCR machine. There are bioinformatics, AI. Through AI today, we can actually have different complementary sequence coding through which we can actually understand nucleotides and study nucleotides and find out the type. Thank you so much.